Next, we are going to talk about hierarchical clustering that we mentioned in the last topic. One potential disadvantage of k-means clustering or GMM is that they require to pre-specify the number of clusters k. Hierarchical clustering is an alternative approach that does not require to have a particular choice of k. It also has an added advantage in that it results in an attractive tree-based representation of the observations, called a dendrogram. Next, we are going to learn about dendrograms and how to perform agglomerative clustering using dendrograms. In hierarchical clustering, objects are categorized into a hierarchy similar to a tree-shaped structure, which is used to interpret hierarchical clustering models. The algorithm is as follows. Make each data point a single point cluster that forms n clusters, where n is the total number of observations. Take the two closest data points and make them one cluster that forms n-1 clusters overall. Take the two closest clusters and make them one cluster that makes n-2 clusters, and repeat these steps until there is only one cluster remaining. Also, calculate the pairwise dissimilarity between each observation in the dataset. First, we must choose some distance metric like the Euclidean distance or the Hamming distance and use this metric to compute the dissimilarity between each observation in the dataset. For a dataset with n observations, there will be a total of n choose 2 pairwise dissimilarities. And then we would like to fuse these observations into clusters. At each step in the algorithm, we have to fuse together the two observations that are the most similar into a single cluster. Repeat this procedure until all the observations are members of one large cluster. The end result is a tree which can be plotted as a dendrogram. To determine how close two clusters are, we can use different methods including a complete linkage clustering, which finds the maximum distance between points belonging to the two different clusters. For example, the orange cluster and the green cluster has the distance d12, which is the maximum pairwise distance between the points belonging to the two different corresponding clusters. Next, we can talk about the single linkage clustering, where we find the minimum distance between the points belonging to the two different clusters. Here, the one plotted in this picture shows the minimum distance between points belonging to the two different clusters. Similarly, there is a mean linkage clustering, there is centroid linkage clustering, and also the Ward's minimum variance method. Depending on the structure of the dataset, one of these methods may tend to produce better clusters than other methods. Dendrogram is a hierarchy of clusters in which distances are converted to heights. It clusters the n objects, each with p features, into smaller groups. Units in the same clusters are joined by a horizontal line. This leaves the most dissimilar points at the top of the dendrogram. The ones at the bottom represent the individual units. This represents a visual illustration of the clusters. Here is a toy example. There are 6 animals based on 5 features. We are going to use the Hamming's distance to create a distance matrix. Hamming distance is the number of 0-1 features that are different. Thus, the Hamming distance between human and ape would be 0 because all the 0-1 features are exactly the same between human and ape. However, between human and mouse, the Hamming distance is going to be 1 because there is one feature that is not the same between the two different observations. In this way, we can create the distance matrix where we can see that the distance between human and ape is 0, while the distance between human and mouse is 1. Similarly, we can create the pairwise distance between each of the observations. Since human and ape are the most similar, we can combine human and ape to form cluster 1. Next, we are going to pretend that cluster 1 is a new observation, and we have to create the distance matrix for this new dataset, which does not have the human and ape, but it has this new observation cluster 1. So we have to find the distance between mouse and cluster 1, snake and cluster 1, and so on. In order to find the distance between a cluster and an observation, we can use one of the techniques like single linkage clustering, mean linkage clustering, and so on. Here we have used the mean linkage clustering, and so the distance between mouse and cluster 1 is 1 plus 1 divided by 2. The distance between mouse and human is 1, and the distance between mouse and ape is also 1, and so the mean of these two is 1. Similarly, the other pairwise distances between cluster 1 and other animals can be calculated. So here's a quick question. Which of the animals has the smallest distance from cluster 1? Well, we can see that clearly mouse has the smallest distance from cluster 1. And so the next step would be joining the mouse and cluster 1 to form a new cluster 2. And then we continue until we have the overall dendrogram. We are going to call the function hclust to perform all the previous calculations and draw the phylogenetic tree. Let us look at an example where the data was extracted from the 1974 Motor Trend US magazine and comprises fuel consumption and 10 aspects of automobile design and performance for 32 automobiles. Here is a summary of the dataset. We have different cars and we have different observations on different features. We are going to use the Euclidean distance to find the pairwise distances between the observations. We are going to use the dist function on the empty cars package using the method equal to Euclidean attribute. The function to perform the hierarchical clustering is hclust. It uses the distance mat and the linkage criterion, which is the average. So here is the cluster dendrogram, and we can see the different cars have been clustered together. And if we draw a horizontal line at height equal to 110, we see that it clearly breaks the observations into two big clusters and one car that forms an outlier, or a third cluster. Here is another function called cuttree. This splits the entire dendrogram into three different clusters. As you can see here, if you split the tree into three different clusters, the one data point here, the Maserati Bora, forms the third cluster, while the other points have been put into two big clusters. To determine how many clusters the observations should be grouped in, we can use a metric known as gap statistic, which compares the total intra-cluster variation for different values of k with their expected values for a distribution with no clustering. We can calculate the gap statistic using the clustgap function used from the same cluster package, along with a plot of the clusters versus gap statistic using the fviz_gap_stat function. I suggest that you can try out these functions on your own and see how they look like.